Hey guys, it's Mrs. Allgood. I am here to teach you about roots today. We'll talk a little bit more about roots on Monday. This is your lecture for Friday. So, roots do three things for the plant. They're going to help anchor a vascular plant into the soil, because remember, a non-vascular plant like a moss really doesn't have true roots. Roots are there to absorb minerals and water from the soil. And then roots can also be for storage. We're going to talk about some storage roots today. So, the good thing about notes today and the notes early next week is that it's going to tell you, we're going to learn about a lot of terms that are part of your plant portfolio. So it should give you a lot more options to be able to go outside and search for some of these things. All right. So the other day we talked about differences between monocots and dicots. Okay. So here we're going to focus a little bit more on their root structure. In fact, that's going to be the main focus today, is the pathway of water transport as it goes through different layers of the root. Okay, so a taproot, like you see on the left there, we find that mainly in dicots and gymnosperms. Whereas fibrous roots, which remember are also called branching roots, those are going to be more common in your monocots, such as your grasses. All right, this second bullet is basically telling you where they arise from, and this may be a little obscure for you because we haven't talked about embryonic roots yet. Taproots arrive from an embryonic root, and it's going to give rise to lateral roots and branch roots. That makes sense since a taproot is a main root, and then you can see up in this picture a little bit that it will have some lateral roots coming off the side. All right, the embryonic root in a monocot dies early on, so you basically have what's called adventitious roots emerge from the stem, and that's kind of why you see all these different roots kind of branch out from the base of the stem. That's why it looks the way it does. A taproot, you can tell by the way it's designed that it's going to penetrate more deeply into the soil than a fibrous root, which is part of a monocot. A fibrous root is going to be a little bit better for ground cover and like preventing erosion. A taproot is going to be able to get to water sources that are more deep in the soil, and that's going to be the advantage there. So a taproot is adapted for growth in soils where the groundwater is not that close to the surface. Whereas fibrous roots, which are your branching roots, which are part of things like monocots and grasses and things like that, they're going to be more adapted where the rainfall is light and doesn't go that deep. But because of the way that those fibrous roots kind of branch out, that's going to make them good for like a ground cover for preventing erosion. Okay, let's keep going. All right, let's talk about absorption of water. So the other day we mentioned a little bit about root hairs. Okay, this is a good picture of a root hair, and you might be looking at that saying, what am I even looking at? This is a seed that has just germinated, that has come out of its seed coat. And so this is what's called the radicle. This is the embryonic root. And so these are the little root hairs coming off. Now, if you have seeds lying around your house, and or even beans, you can do it with beans as well. They just can't be from a can or frozen. But what you can do is you can soak those beans or seeds in water for about 24 hours, and then you can drain the water off and wrap them in a wet paper towel, and they will germinate on their own. Okay? So we'll be talking about seed germination a little bit next week, but I wanted to give you a better idea of what those root hairs are like. They are there to increase the surface area to help with absorption. So that's what that little bullet says right here. The root hairs are going to increase that surface area for absorption tremendously. Root hairs are an extension of an epidermal root cell or a root epidermal cell. They are not a lateral root. Okay, lateral roots come from a different layer. They don't come from the epidermis. So as we get a little further into notes today, you'll see that. All right, let's keep moving. Okay, so we're going to look at evolutionary adaptations of roots. So on your plant portfolio list, for some of these, I don't know if you'll be able to find any of these, but you might be able to find some of them. It'll say, if it said something like a pneumatophore, which we'll talk about in just a second. Well, what kind of modification is it? If it says something like what kind of modification is it, it's asking you if it's a modified root, stem, or leaf. Okay, so today we're talking about modified roots. On Monday, we'll talk more about modified leaves and stems. So these are called prop roots. Okay, we kind of always expect roots to be below the soil. They're not always. Prop roots do what their name implies. They are there to support the stem. We tend to find it more in some tropical plants where the soil's just not as good and the plants are a little more top heavy, so they need that extra support. It can also be where the soil is a little more sandy and shallow and unstable. So this wasn't a picture I took. I couldn't find mine, but this was similar to one I took when I went to a corn maze a few years ago. There's amazing things, even though you've eaten corn maybe your whole life and maybe you've shucked corn. There's always neat little things that you see if you go to a farm and you see the corn growing. You'll see trichomes, you'll see parallel venation, you'll see all kinds of things that you've learned about already in this unit. And one of the things you'll notice that corn indeed does have prop roots. Okay? Now, I don't expect you to go to a farm and find a corn plant. I think that would be a hard thing to find this time of year. So I don't expect you to find a lot of these modified roots. There might be one or two that you might be able to find. Storage roots. This is one you can find. You can find this at the grocery store. Storage roots are roots that may store food and water. So a beet is a storage root. A carrot is a storage root, but realize that a carrot is also a taproot. So make sure you look on your plant portfolio list. I want to say I'm letting you use carrot as a taproot, but not as a storage root or one or the other. In other words, you can't use a carrot for both. I wouldn't just assume that something is a storage root because it grows underground. Some things, as you'll see next week, are actually modified stems. So beets are storage roots, radishes, turnips. If you're not sure, I would just look that up to be sure. But this is something that you can find and take a picture of. Pneumatophores. Pneumatophores are kind of cool. You'd have to really be a little more adventurous to go find these, and I don't know that since we're under quarantine that you'll be venturing out to any swamps anytime soon. Pneumatophores are also called air roots. We see these pretty common in your swampy trees, near mangroves, which we often find in the swampy areas. Basically, these are extensions of the roots that come up out of the water, and they often come to like a point. Oh, hold on, my dog. Okay, guys, sorry, I'm back. So these are actually like extended pieces of the roots that are coming up to get oxygen because the mud is too thick and the oxygen can't get to the roots. Okay, now if you see something like this, this is not a pneumatophore. Pneumatophores generally are pointy at the top. This, if you see, kind of resembles a knee, so this is called a cypress knee. This is not a pneumatophore, so don't include this in your plant portfolio. Buttress roots. Now, let me do my disclaimer for this. I know that trees have extensions of their roots that can start above the soil. This is a special type though, and you're probably not going to find it in North America. So please don't take pictures in your backyard and tell me it's a buttress root, because it's probably not. You are probably, unless you've been to somewhere tropical recently, which I doubt you've been there anytime in the recent past, given the coronavirus, this is really just going to be found in tropical locales. So these are aerial roots that look like buttresses. Buttresses are an architectural feature like you see here. Again, it's for support. So, but these are thin projections, okay? You might see some kind of root of a tree starting above the soil. A lot of trees have that. These are a very specific type, and they typically only grow in tropical locales. Okay, strangling aerial roots. Let's take a look at this just a little bit at a time. This is when seeds of the strangler fig germinate in the branches of taller trees of another species, and they basically send their roots downward. Now, supposedly there's a tree like this. Hold on one second. All right, so we were talking about the strangling aerial roots. So I've heard that there is a tree like this in downtown Greenville near the Governor's School. So you might be able to find a picture that's kind of like the one I'm showing you here. But basically the seeds of the strangler fig germinate pretty high up and they sort of send roots downward. So they send numerous aerial roots down to the ground. The aerial roots wrap around the host tree, and the host tree eventually dies because of being all covered up. So those are your 5 types of modified roots. Okay? Okay, now we're getting into the technical. What we are looking at now is the layers in a cross section of a root. So I want you to imagine that we take a root and we slice it sideways to do a cross section, and then we put that cross section under the microscope. This is what we'd be looking at. And so basically in this section of notes, we are tracking the pathway that a droplet of water would take as it enters the outside of a root, maybe through a root hair, to when it gets to the xylem. Okay, because once it gets to the xylem, we know that tracheids and vessel elements will transport it upward in the plant. Okay, so that's what this section notes is all about. We are learning about the different sections of a root as if we were looking through a cross section, and we are also tracing the path that a droplet of water takes as it comes through the root. So from outermost layer to innermost layer, if we're looking at a root cross section, okay, the outermost layer is going to be your epidermis. So if we look at it here, see if I can get a little color on this. The epidermis is just one layer. So it's just that. Okay, and notice you see some little root hairs coming off. Okay, the root hairs, remember, are extensions of epidermal cells. So they're not going to come off of every cell, but epidermal cells, some of them can have root hairs. Okay? Now, remember, this epidermis is just what I colored in yellow. It is just one layer thick. The cortex makes up the bulk of the root. It is where starches can be stored away, and it is made of parenchyma cells. So I want to show you what that is in this picture. So in this picture, the cortex is basically this part in through here. This is the cortex. And if you remember what we said parenchyma cells look like the other day, that's what they look like. All right. Next is the endodermis. Okay, so the epidermis is on the outside. Then we have the cortex. Then we're going to have the endodermis. The endodermis is the innermost layer of the cortex. So technically still part of the cortex, just the innermost layer. So this is a really important topic here. We sort of have to take a little detour to talk about this suberin. So the cell walls of the endodermis contain suberin, which is a waterproofing substance. And that layer of suberin creates a water impenetrable barrier called the Casparian strip. Now I know what you're probably thinking. Aren't we tracing the pathway that a droplet of water takes as it enters a root? Why would we want a layer that is impenetrable to water? We don't want to make the root completely impenetrable to water, but we do want to make the water follow a certain pathway. So it would be kind of like if you were waiting in line for a concert, okay, that they might have certain barricades set up so that you would have to get in line in a certain place and you couldn't just line up anywhere. Okay, that's kind of the purpose of this Casparian strip. So why do we have this Casparian strip? It's going to prevent water from seeping between the cells to get to the vascular tissue. In other words, it makes water have to go through the endodermal cells. So I'll show you in this picture in just a moment. All right, so this is kind of what we're getting ready to talk about. Let me pick a color. Okay, so there's basically two pathways that a droplet of water can take as it comes into the root. So this droplet of water can come in and it can enter the epidermis. Okay, so it can come through here and then it can come through the plasmodesmata and pass through cells of the cortex, and then it can pass through more plasmodesmata and get into the endodermis is only one cell layer. This is only one cell layer. This cortex is probably several cell layers thick. But anyway, it can come through here. It can go through another layer called the pericycle, and then it can get in the xylem and then be transported upward. Okay, that's kind of like the normal pathway. All right, there is another pathway though. Okay, so let me change colors a little bit. All right, some water droplets are going to try, I don't know what I'm going to say, they want to take a shortcut, but they're going to try and take a different path. So they're basically going to travel through the cell wall. So notice what this little red line is doing. It is traveling through the cell wall, but look, now it hits a barricade. It hits that Casparian strip, and it can't take this little shortcut anymore. It can't just travel through the cell wall. So that is going to force it to actually enter the endodermal cell. And then it's got to travel the rest of the way within a cell instead of just traveling through the cell membrane. So I hope that that makes sense. So we're going to take a closer look at that on the next page. So just like I showed you on the last slide, there are basically two pathways. The pathway that I showed you first with that darker blue line was the intracellular route. when it was traveling basically entirely within the cytoplasm or within the plasmodesmata that connect the cytoplasm of adjacent plant cells. So in the Symplast pathway, water crosses the cell wall and the cell membrane of epidermal root hairs, because that's what it's typically coming in through. And it basically, in the Symplast pathway, it's going to enter the cytoplasm right away. So this is the blue line in this picture. This is the Symplast pathway. Okay? It enters, it crosses the cell wall, and then it's going to travel within the cytoplasm and pass through plasmodesmata. So here's your epidermis. Okay, it's in the cytoplasm. It's going to go through plasmodesmata and then basically travel within the cytoplasm of cells of the cortex. Then it gets to the endodermis and then to the xylem. Okay, so it's going to travel from the epidermis to the cortex to the endodermis. There may be another layer in here called the pericycle, which is not shown here, and then it will enter the xylem. Okay, so this Symplast pathway is the blue pathway that you're looking at in this picture. Next is the apoplast pathway. So this is the pathway that I showed in maroon before. This is where the water droplet is mainly traveling within the cell wall, and it doesn't enter the cytoplasm of a cell unless it's forced to, which it will be by the Casparian strip, which is like a blockade. It would be like if you were going to wait in line for a concert and you thought, oh, I'm going to get in line over here. I'm going to get in line over here. And then we're going to sneak in and we'll cut in front of everybody. Eventually, there's going to be a blockade where you can't take that shortcut anymore. And you're just going to have to get in line with everybody else. So in this pathway, the water is moving through the cell walls, but it doesn't actually enter the cytoplasm of the cell until it gets to the Casparian strip. And so the Casparian strip is made of suberin, is what's forcing it to enter an endodermal cell, a cell of the endodermis. So if we look at this red colored line here, or pinkish colored line, whatever color you see that as, notice it enters, but it doesn't actually go into the cytoplasm. It's going to enter and travel within the cell wall, travels within the cell walls, and notice when it gets here, this black is the Casparian strip that is in between endodermal cells in the cell wall. So the function of that Casparian strip is to force that water droplet to now enter the cytoplasm of an endodermal cell, and then it'll go through a layer called the pericycle, and then it can enter the xylem. So the bottom line concerning apoplast and Symplast pathways, basically no matter which pathway water and solutes take, they must eventually cross a cell membrane and get into a cell at some point before getting to the xylem. They just differ at which point they enter a cell. Because in the Symplast pathway, which is blue, they enter the cytoplasm right away, and they travel through cytoplasm to cytoplasm by going through plasmodesmata. In the red pathway, which is the apoplast pathway, they basically just travel through the cell wall. They have no intention of ever entering the cytoplasm of a plant cell. But once they reach that Casparian strip, which is part of the endodermis, that is forcing them into the endodermal cell, into the cytoplasm, and then it can go the rest of the way within a cell. Okay, so far, in tracing this droplet of water, it enters the epidermis, then it passes through several layers of the cortex, so epidermis, Could be coming in through a root hair, but that's still part of an epidermal cell. It's going to pass through several layers of the cortex, and then it's going to enter the endodermis. So that's the point that we're at, but we need to go further inward than that. Okay, so before you start filling in your picture, let's fill in here. All right, then we're going to get to the stele, which is also called the vascular cylinder. So in the plant unit, when you see that term vascular, you want to think xylem and phloem. Now, the outer layer of the stele, or the outermost layer of the vascular cylinder, is called the pericycle. And it is made of parenchyma cells, and this is the layer that can give rise to lateral roots. So if you go back and look at one of our taproot pictures, it shows lateral roots coming out of it. That comes from the pericycle. So I want you to fill in your blanks there, and then I'll talk you through this. But go ahead and take a moment to fill in your blanks. All right, so we want to start on the very outside layer and work our way in. All right, your epidermis. Your epidermis is this layer right here. That's your epidermis. Okay. Your cortex. Your cortex is from here to there. Okay, all the way around. That is your cortex. The next layer in that you should encounter, let me see if I can pick a different color here, should be your endodermis. Okay, so this is your endodermis here. Now really, this black bracket really should include that. Okay, the endodermis is the innermost layer of the cortex. The next layer that you're going to encounter is going to be your pericycle. Okay. Notice what we bracketed here. This entire thing is your stele or your vascular cylinder. Okay. Not really including the endodermis, but including the pericycle. So your pericycle is going to be your outermost layer of the stele or the vascular cylinder. The larger circles here are going to be your xylem. The smaller circles that are kind of more external to your xylem, those are your phloem. They're kind of located in between the bigger xylem circles. And then the core is going to be your pith. Now this picture over here is just showing you that your lateral roots, this is a lateral root, like you might see associated with a taproot, it will come out of the pericycle. So let's talk about the arrangement of xylem and phloem. Okay, xylem and phloem are arranged differently in monocot and dicot roots. So you got to get these two down pat before we start talking about secondary growth in stems, and then you got to learn the way xylem and phloem look in a monocot and dicot stem. So we're taking this a little bit at a time. We're just doing the roots today, but you got to make sure you really pay attention to this. So let's take a look at our picture. Monocots are going to have a pith in the center of the root. and that is just made up of parenchyma, which is ground tissue, parenchyma cells, which make up ground tissue. Okay? And then they're going to have a ring of xylem and then a ring of phloem outside of that. Pretty straightforward. Dicots are a little different. Dicots, if you look at my cursor, xylem is here. It kind of makes like a T or an X. And then the phloem is what I drew in purple here. That's your phloem. So xylem here, phloem are in between the spokes, if you want to think of it that way. There is no pith in a dicot root. Pith is in the center. There's no center here. The center has xylem. So there's no pith here. All right. So another thing, I don't know if I gave you this slide or not, but you can find these kind of images on Google. It's out there. I would practice labeling my layers. Okay. Notice that the vascular cylinder is what we'd call this area, how much smaller it is in a dicot root versus a monocot root. Okay, they're not the same size. This is the vascular cylinder in a monocot root. This is the vascular cylinder in a dicot root. This is way smaller. It kind of forms this X-like pattern. This, you have a pith, and you have your xylem and your phloem. So I know that this is a monocot root. I know this is a dicot root. Okay. So I would, if this were me, I'd be grabbing some colored pencils. It's hard for me to draw with my little finger on the mouse pad here. So I'm going to let you pause this. I would pause it. I would draw it and color it. I would be drawing it with you, but that's really hard for me to do using the mouse pad. So pause this, draw it, and then we will resume in just a moment. Okay, so let's talk through this together. It might be helpful to start on the inside and work your way out. This is your monocot root. I didn't label it, but your pith would be in the center. The green ovals that you see here would be your xylem. Your phloem would be smaller and would kind of be interspersed, but more external to the xylem. The purple is going to be your pericycle. That is the outermost part of your vascular cylinder. Then you're going to have your endodermis. Then you're going to have your cortex. Then you will have your epidermis. Coming over here to look at the dicot root, I expect my xylem to make an X-like formation and for my orange phloem here to be in between the spokes, okay? No pith. Then I have my pericycle, then I have my endodermis, then I have my cortex, then I have my epidermis. Okay, and so this is just kind of thinking big picture. This is the note I believe we're going to end on today. Yes, this is your last slide. Longer lecture today, sorry. So from the soil, water typically gets taken in through root hairs, which are extensions of epidermal cells. Okay, from the epidermis, we know it's going to pass through the cortex, and then through the endodermis, and then through the pericycle, and then to the xylem. Okay, xylem, remember, consists of two specific kinds of cells, probably one more so than the other, depending on what kind of plant we're talking about, but those two types of cells are tracheids and vessel elements. They're going to take water up the stem to the veins of a leaf, to the parenchyma cells, okay, could be to the palisade parenchyma, could be to the spongy parenchyma, and then the water vapor will exit the air spaces of the leaf. It will leave through the stomata and go to the atmosphere. So one of the things I want you to think about here is water is not getting pushed up the stem, it's actually getting pulled. So the fact that water vapor can be lost through the stomata of the leaves and that the water is going to be exiting, that is the force driving the pull of water up the stem. So as more water leaves as water vapor through the stomata, more water is going to be pulled up through the tracheas and vessel elements up the xylem. That is the driving force. All right. That's been roots. So please do the corresponding work. I'll clarify that on either Google Classroom or It's Learning.